Hello, everyone. This is Frank Ambrosio, bioinformatics scientist with Theogen Genomics. Welcome to the second session of the Bioinformatics Fundamentals training course. Today, we are going to be talking mostly about phylogenetics, genomic epidemiology, and all of the concepts that we need to understand in order to be able to apply genomic epidemiology in a clinical setting, in an outbreak setting, and in a genomic surveillance setting. So I'm going to share some slides just to organize my thoughts, and we can jump right in. Okay, again, I am Frank Ambrosio, bioinformatics scientist with Theogen Genomics, and this is the second session of the Bioinformatics Fundamentals course offered by Theogen. So the main objective of this course is to introduce these concepts of bioinformatics, next-generation sequencing, and genomic epidemiology so that we can understand how best to apply these concepts in the field of pathogen genomics, whether that be in a clinical setting or a public health setting. So this is session 2. Today we're going to be talking about the fundamental concepts of genomic epidemiology. We'll be going over how phylogenetic trees are constructed and how to use them in both an outbreak investigation and a genomic surveillance context. And we'll be reviewing a few case studies on this subject. Genomic epidemiology. What is it and how do we use it? So epidemiology is the study of the propagation of diseases through populations. And we have traditional epidemiological techniques such as contact tracing, which allow us to visualize the propagation of a pathogen through a community. And in that way, we can make informed decisions about public health policy. What genomic epidemiology does is it combines the use of pathogen genomic data with the traditional epidemiological data in order to get a higher resolution map of the transmission network of a pathogen through a community. One of the first steps of genomic epidemiology involves characterizing our samples. So first, we need to know what taxon our sample belongs to, what is the species, and then other characteristics of the pathogen are also very important. For example, subtyping. So with SARS-CoV-2, we saw lineage distributions changing throughout different regions in the United States. We continue to monitor this to this day. If you are interested, the CDC Nowcast page has a great visualization of the variant distribution across all HHS regions. Another characteristic that we want to understand about our pathogens before we start creating a tree is the AMR gene prevalence. Which samples have antimicrobial resistance, which samples don't, and is there any commonality? Is there a trend? Do we notice that samples are clustering together that happen to have a particular AMR gene? Are we seeing samples that are completely different species that have the same AMR genes? This will all inform our genomic epidemiological investigation. And then another version of subtyping, MLST, multi-locus sequence typing. This is a concept that we cover in the first session of this Bioinformatics Fundamentals course. So if you're interested in learning more about MLST, go check out that course for sure. But for now, what we want to understand is which of our samples belong to the same sequence types. And belonging to the same sequence types means they have the same version of certain genes within their genome that could be informative for a genomic epidemiological investigation as well. Patterns in these data act as indicators of potential outbreak events. So we look at related samples that could all be part of the same outbreak. And from there, we can use the genetic relationships between these samples to try and pinpoint things like introduction events, instances where we saw a pathogen rapidly mutating. There might be some environmental pressure on that pathogen. So we look through these data in order to understand the dynamics of a pathogen spreading through a community, whether that community be a hospital or a dorm facility or an entire nation. So phylogenetic analysis for outbreak investigations is slightly different than for genomic surveillance activities. With an outbreak investigation, we're using the evolutionary relationship of a set of samples as a proxy for transmission. So the more closely related samples are genetically, the more likely it was that there was a direct transmission event between these two samples. And typically, based on shared SNPs between core groups of samples in an outbreak investigation, we can identify the directionality of these transmission events. We can infer the directionality. from the accrual of new SNPs over time, new single nucleotide polymorphisms in the genomes over time. So sequencing allows this high resolution understanding of the genetic relatedness, though it is not to be misunderstood. This is not a replacement for traditional epidemiological techniques. It is something to be added to the traditional epidemiological techniques. So we have the high resolution genetic relatedness, and we have all of the other factors that we normally take into consideration in an outbreak investigation, such as proximity to other individuals who have the same type of infection, if they're living together, working together, eating together. These are all potential outbreak loci. They're potential super spreader event loci. So we must take everything into context when performing this type of investigation. There are many approaches to performing phylogenetic analysis. There are many algorithms for phylogenetic tree construction. These will all be informed by the upstream bioinformatic processes such as genome assembly, read cleaning. Everything upstream of the tree building will impact the topology of the resulting phylogenetic tree. So everything needs to be taken into consideration, not just the phylogenetic tree building algorithms, but all the various combinations of upstream tools that could be contributing to the ultimate topology that we observe in our phylogenetic trees. So for genomic epidemiology, the key takeaways are going to be in genomic epidemiology is the practice of incorporating the sequencing data into our traditional epidemiological investigation techniques. By combining these techniques, we get a 1 + 1 = 3 effect, meaning that we get way more by combining these techniques than we would with either one individually. Genomic epidemiology is useful for both pathogen surveillance at a larger scale, placing our samples in context, and also outbreak investigations, visualizing and observing the propagation of a pathogen through a population. And of course, using genomics allows a high resolution image of transmission events. We can say with more certainty whether or not there's been a transmission event between any two individuals in the network. And that gives us a lot more confidence in our transmission network analysis. By using nucleotide level assessment of genomic relatedness, we can use that genomic relatedness as a proxy for transmission events, and that is the process of phylogenetic analysis. So let's learn some more about phylogenetic analysis. Here we are assessing the evolutionary relationship between a set of samples. So we're all familiar with the concept of a family tree. Phylogenetic tree is a family tree based on the unique genomic signatures of each sample involved. The more closely related genetically any two pathogens are, the more likelihood that they were linked by a transmission event between two individuals. If they're genetically identical, there's a very high likelihood that one individual transmitted a pathogen to another and it has not evolved. That is rarely the case. Typically, we see the accrual of single nucleotide polymorphisms over time. So here is a good point to introduce the most important question here, the question that I get asked most often during phylogenetic analysis of outbreak investigations. What is the threshold of single nucleotide polymorphisms that qualifies a transmission event? How closely related do two samples need to be before we consider them a likely transmission event? And the answer to that is not something anyone ever likes. It depends. It depends on a number of factors, and three factors that play the biggest impact if I had to do a principal component analysis here. The first up would be the evolutionary rate of the pathogen involved. Some pathogens are highly clonal. Mycobacterium tuberculosis evolves very slowly. Other pathogens evolve very rapidly. And even within the genome, the rate of evolution, the rate of mutation across every nucleotide in the genome is not even. So the first thing we must consider is the evolutionary rate of the pathogen in question. The next is the time scale of the outbreak. If the outbreak has been going on for a year, it will likely have much more diversity in the genetic profile of all the samples in the outbreak than an outbreak going on for 2 weeks. And that just makes sense. And then finally, the breadth of the outbreak. How many individuals are infected? The more individuals, the more diversity you're going to see in your genomes. So while we're using the evolutionary relationship as a proxy for the likelihood of transmission events, it is an imperfect science there. We must take into consideration many factors when defining that threshold of a likely transmission. And again, many approaches have been developed to perform this. One of my favorites is to use the microbe trace platform. It's a web-based platform where you can take phylogenetic trees and visualize them as transmission networks, and then try out a number of thresholds of transmission. So if that pathogen only accrues 5 or 10 SNPs per transmission event, you'll be able to visualize that in a tool like microbe trace. So in general, the process is the same, but the algorithms underlying these processes tend to be slightly different. So the math will be different, but in general, the first step for phylogenetic analysis is to identify the number of genetic differences amongst a set of samples. Now, when I say this, there are many different ways to identify genetic differences. I'll give 2 examples. The first example is going to be a reference-dependent genetic difference algorithm. The tool LiveSet takes a reference genome, so a known genome that is related to all of the pathogens in your sample set. I'll use Mycobacterium tuberculosis as another example here. So we are measuring the genetic differences relative to a reference genome. So every sample will be compared to that reference genome, and all of the genetic differences relative to that reference genome will be counted. Now, another approach is something like KSNP3, where there is a reference-independent methodology for identifying genetic differences, where we do a pairwise comparison of every sample in our data set. So we see all of the genetic differences between each sample and each other sample in the data set. So these are 2 different approaches to getting at the same concept. We're looking at the genetic relatedness between our samples. From there, once we have our genetic relatedness, typically in the form of what's called a SNP matrix, a single nucleotide polymorphism matrix, where we have a pairwise comparison of all of our samples and the genetic distances between each of them, we then take those distances and we use that to infer the evolutionary relationships. And the concept here is very simple. Samples with fewer differences are more likely to be closely related than samples with many differences. From there, a tree is constructed. That tree file, typically in Newick format, which is a standard bioinformatics format, can then be visualized on a number of different phylogenetic tree visualization tools, such as iToll, Fandango, Microbe Trace, and there are many more. Many ways to define genetic differences. I should also mention SNPs are not the only way. Single nucleotide polymorphisms are not the only way to define genetic differences. We can look at something like a whole genome sequence typing, where rather than looking at individual nucleotide differences, we're looking at whole different versions of genes and constructing our phylogenetic tree based on having the same sets of genes, having matching sets of genes within genomes rather than looking at individual nucleotide differences. So all of this should be taken into consideration and well understood by the genetic epidemiologist, by the genomic epidemiologist, when investigating an outbreak, when looking at a phylogenetic tree and interpreting that tree to inform public health decision-making. So defining genetic differences, how is this actually done? This is a nice simple example here. What we're looking for are single nucleotide polymorphisms in this case, but you could imagine core or whole genome multi-locus sequence typing. In the case of SNPs, each of these colored dots would be a single nucleotide polymorphism. So a single nucleotide that is different from a reference or any of the other samples. When working with core genome or whole genome multi-locus sequence typing, each of these colored dots would represent an entire gene. So the difference between C and B and A here would be an entire different version of a gene at this loci with the core genome and whole genome multi-locus sequence typing approach. But again, with SNP approaches, we're looking at single nucleotide differences. So how do we get from the differences between our genomes to a phylogenetic tree? Regardless of whether they are SNPs or entirely different alleles or versions of genes, we want to construct a phylogenetic tree where more closely related samples appear next to each other in the visualization. And there are many algorithms to accomplish this, but let's break it down just in a very simple example here. So what are all the different tree building approaches? There are many. If we go back one slide, the really simple example here is if you have one mutation that is common among all of your samples, that is the reference, that is the root sequence of the phylogenetic tree. From there, we look at shared SNP groups. So we see 3 genomes here all have this orange SNP, and we can see they are all clustered together on this tree. Similarly, we can see this teal SNP here is represented in these 2 genomes here. And then finally, this red SNP is unique to genome C. The green SNP is found only in genomes A and B, and this darker red SNP is found only in gene E, or in sample E. So we can see that over time, most likely what happened based on, or the maximum likelihood of what happened is that we have all of the genomes with this blue SNP, so that likely evolved in this set of genomes first. From there, there was a common ancestor, and these are inferred nodes. What inferred nodes are, we don't actually have the sequencing data on the sample that is the common ancestor of these 2 clades. We just don't have that. But we can infer there was a common ancestor based on the shared mutation at this blue loci. Based on that shared SNP at that loci, we can infer that there was an ancestral sequence that did not have any of these other SNPs. It just had the blue ones, and the rest of the SNPs occurred after that. So that concept there is really how all phylogenetic tree building algorithms build trees. They understand that SNPs that are shared by all of the genomes in your data set are more likely part of the ancestral strain. And from there, they infer these other genomes, these ancestral strains that must have existed for these subsequent strains to have evolved. So you can have different tree topologies emerge based on different algorithms employed. That's really important to understand because there could be very subtle, very nuanced differences that could result in completely different tree topologies. This is, again, a very simple example, just for illustrative purposes. Under the hood, phylogenetic trees are performing a much more complex mathematical process here, but suffice it to say that SNPs that are more common occur earlier on in the phylogenetic tree. So let's just touch on several tree building algorithms. Just for awareness, no one needs to actually understand the math behind all of these unless you plan to start implementing some of these, which if you do, please reach out. We would love to work with you. So parsimony trees. This algorithm attempts to minimize the number of changes in the phylogenetic tree, the total number of changes. So what is the easiest way to get from point A to point B? Don't infer any additional changes, just keep it as simple as possible. The distance tree, this is going to be pairwise distances and nothing else. This is a very simple relationship. This is exactly what I was showing earlier. So if we were going purely on distance, that is the algorithm that I went through in that very simple example earlier. Maximum likelihood, this is the most common tree building algorithm employed today. It is a statistical approach. It tries to find the maximum likelihood of the observed SNP changes. So that would take more into consideration what are the shared groups of SNPs? What is the most likely next SNP to emerge? If SNPs are found in relatively the same amount of samples in a data set, this algorithm will attempt to figure out which one came first and which are related to one another based on the relationships between these SNPs, based on what was most likely. And then, of course, we have Bayesian approaches. These are computationally intensive approaches that try to predict the posterior distribution based on a prior distribution, Bayesian approaches. If you're interested in reading about any of these, there is infinite materials online. But I just want everyone to be aware that there are multiple different algorithms. They do different things under the hood. And now that we've been exposed to these algorithms, we can just be informed. Which algorithm was used to build this tree is a good question to ask. So once we've constructed our phylogenetic tree, we can visualize it in one of many phylogenetic tree visualization softwares. And then comes the hard part, interpreting the phylogenetic tree and using that interpretation to inform public health decision-making. So let's go over the anatomy of a phylogenetic tree, just so we're on the same page as far as terminology. So we have our terminal nodes. That is, every letter here is associated with a terminal node of the tree. These are also referred to as tips or leaves occasionally. And these are the known samples. These are the samples for which we have sequencing. We have taken specimens from individuals, performed sequencing, and constructed a phylogenetic tree. These are the nodes that end the tree, so there are not going to be any sublineages from a terminal node. Next up, we have these internal nodes, and these are inferred. So these, we don't actually have any samples. We don't actually have any sequencing data for these. We can infer their existence based on the shared SNPs of all samples within that clade. So that we infer the existence of a common ancestor. And here we can see a few of these examples highlighted in yellow. Next, the length of a branch of a phylogenetic tree can represent a few different things. Typically, this is representative of genomic distance. So samples that have a longer branch length separating them, they are more genomically distantly related. Samples with a vertical line connecting them are genetically identical samples. So here we can see samples A and B are genetically identical. To compute the distance between the AB clade here and sample C, we would add the genomic distance horizontally from right to left, and then add the genomic distance horizontally from left to right. And that would be the total genomic distance between these samples and these samples. And to illustrate that, both samples have this one SNP. Samples A and B gained this green SNP, sample C gained this red SNP. So if you count 1, 2 SNPs, if this branch length is 1 and this branch length is also 1, clade AB differs from sample C by 2 SNPs. And here we can see near zero samples or zero distance samples will be represented by a vertical line here. That is very rare to have in nature, to have genetically identical samples. But sometimes the algorithm, the distance is so small relative to the distances between other samples, they will appear identical here. And again, just to reemphasize, the genetic distance between sample C is the combined distance between clade AB, the common ancestor, and then the common ancestor and sample C. So branch length is typically the genomic distance. However, it can also be the time of emergence. So there are algorithms that compute branch length based on the time of collection or the time of sequencing or the time of submission. And here that would be a more temporal phylogenetic tree than purely a genomic distance phylogenetic tree. So the term cluster gets thrown around a lot. Essentially what cluster means is samples that have a close evolutionary relationship. What does close mean? This goes back to the question I posed earlier. What is the threshold? What is the genomic diameter of a cluster? What do you set that threshold at? And again, it's going to be based on the same 3 things: the evolutionary rate of the pathogen in question, the number of individuals that are part of the outbreak, and the time scale of the outbreak. All of these things are going to inform what you set that threshold at. And really, you won't have a good idea of what makes sense as a rational threshold until you've taken a look at the phylogenetic tree of your samples. So once you see the general distribution of genomic distances between all of the samples in your data set, and you see that some of them are clustering close together and some of them are not, you can start to see where that threshold is best set. For example, in this very simple example, we can see that we have 2 identically related samples here, and we have 2 very closely related samples here. If we were to set the genomic threshold for a cluster at 2, we would have a cluster up top of these 3 samples here, and these 3 samples differ from these 2 samples down here by 1, 2, 3 for D and 4 for E. So if we set the threshold at 2, we set it along here, we would get nice, clean, 2 distinct clusters there. And that threshold is informed by looking at the phylogenetic tree and looking at the general distribution of genomic distances among all of our samples. So in this instance, sample A and B make up a cluster. If you set the threshold to 1, samples A and B would be the only cluster here. D and E, depending on whether it was 1 inclusive or exclusive, would also be a cluster. So here we have another cluster of samples D and E, and I just want to point out that if you were to set the threshold to 2, we would have totally different looking clusters here. One thing to be aware of is that we are not including the inferred nodes in these clusters, as that would make everything much more messy since we don't know for sure that those samples actually exist. Alrighty, so just a recap of the terminology. The terminal nodes, these are the samples that we perform sequencing on. We have their genomes, and they are how we constructed the phylogenetic tree. The internal nodes, these are the inferred common ancestors. We don't know if they actually existed. Multiple SNPs can be introduced at the same time. So we cannot say for sure that these inferred common ancestors actually exist. Branch length is a visual representation of the genomic distance or the time of emergence, both of which are informative under different contexts. And cluster is a general term referring to groups of samples with a close evolutionary relationship. What does close mean? This is going to really depend on the context of your outbreak or surveillance activity. So the key takeaways here are going to be, we're using evolutionary relationships as a proxy for a transmission event. Close evolutionary relationships, more likely that there was a transmission event. There are many different approaches to constructing a phylogenetic tree and many more approaches to all of the steps leading up to the phylogenetic tree. And any combination of those approaches should be viewed as a unique methodology. So it's best to understand what methodology you're employing by the time you get to a phylogenetic tree. So what does that look like? You want to know what the laboratory process was for extracting the genomic material from your sample. You want to know which library prep kit was used to prepare your samples for sequencing. You want to know which sequencing platform and flow cell was used. Then you want to know what process of read screening, read cleaning was used. Was quality trimming, were reads removed? What was your quality threshold for those reads? Then what assembly algorithm did you use? Was it a conservative or a more aggressive assembly algorithm? What were the parameters of that assembly algorithm? Then you have your genomes. Once you have your genomes, how did you compute the evolutionary distances between these genomes? And then what algorithm did you use for phylogenetic tree construction? So you can see any unique combination of all of those steps should be viewed as an individual unique methodology. And I'm sure you can find papers benchmarking different combinations of those. I know for sure there are benchmark papers comparing different tools for each of those steps against one another. So make an informed decision and definitely understand what tools led to the phylogenetic tree that you're attempting to interpret. So now we have a few case studies in public health. This is a nice overview of just a few examples of how genomic epidemiology has been used in a public health setting. That being said, a couple of these public health settings are also clinical settings, so this is in no way public health specific. So starting with SARS-CoV-2, we should all be familiar with SARS-CoV-2 at this point. It's routinely sequenced and characterized by bioinformatics workflows. Genomes are assembled and then phylogenetic analysis is performed for millions of samples. You can go check out a number of sites. I know taxonomy.org has a great phylogenetic tree that's virtually infinitely zoomable. NextStrain is one of the best places to visualize SARS-CoV-2 phylogenetic trees. NextStrain is a fantastic tool. If you have a Newick file or a SNP matrix, you are ready to go with NextStrain. Now, it uses a SNP calling approach and a maximum likelihood tree building algorithm. So that's important information to know. Usher is another version where they use maximum parsimony for phylogenetic placement, also based on SNP calling. These are two examples of different tools that have different underlying tree building algorithms, but essentially the same genomic difference computation style. So they're both calling SNPs, but then they use different tree building algorithms. So here we have an example of a NextStrain tree. And this is a real world example where we had a number of samples appearing in Washington in the United States. And we had a genomic relationship to samples from outside of the United States. And we were able to conclude that there was likely spread of SARS-CoV-2 in the Seattle area based on this phylogenetic tree here. And again, this is a very simple phylogenetic tree, but what you can see is a vertical line relating these three samples and a genomic distance of 1 relating to this fourth sample here. So if we were to set the genomic distance threshold at 2, we would have a unique cluster up here in the top and one unique cluster down here. Now, this appears to be a unique introduction event unrelated to these other samples. However, if we were doing a more genomic surveillance-based analysis here, we would say, okay, how many SNPs have accrued between these samples here and these samples here? It appears to be 1, 2, 3, 4 SNPs difference between these 3 identical samples and this one sample down here. Now, that's not a lot of SNPs difference. This may not have been a direct transmission event. However, you could see how there is likely a relationship between these samples given the relatively low number of SNP differences between them. And with that information, what you can do is you say there's a unique introduction event in the Seattle area. There has been community transmission in the Seattle area, and the introduction event may have come from wherever the individual with the genomically related SARS-CoV-2 sequence is from. And this is a very simple example, so let's take a look at a more complex example. And everyone's bored of SARS-CoV-2 at this point, so I thought I'd use something that's an emerging pathogen here. We have the increased prevalence of Candida auris in southern Nevada. This has been a pretty big issue over the past year or so. There are 2 simultaneous outbreaks of different clades of Candida auris causing outbreaks in southern Nevada currently. So sequencing and characterization was performed on all of these samples, and phylogenetic trees were constructed. This allowed us to identify highly related samples using high-resolution genomic epidemiology. In addition to assembling the genome, additional characterization analyses were performed, the first of which is clade typing. We wanted to know which Candida auris clade did these samples belong to, and then antifungal resistance gene screening. Did they have any mutations that look like antifungal resistance mutations? So after performing that analysis, we performed SNP calling, and then we were able to construct our phylogenetic tree using a maximum likelihood approach. And here is the tree that we produced. Now, the first thing we wanted to know is this Nevada group, is it related to any of our other samples? Is it likely transmission? Is there a likely transmission event from any of the other known ongoing outbreaks to our Nevada group? So if we just take a minute to orient ourselves on this tree, we have 2 New York outbreaks that were ongoing and well studied, lots of sequencing going on there. We also had a mountain group that was genomically distinct from the New York groups. And even within the New York groups, there is a separation of 2 clades, 2 clusters of samples. And now we take a look at the Nevada group in context with the rest of our samples here. So rather than just sequencing and constructing a tree from our Nevada group, we placed our samples in the larger national context by including genomes from previous outbreaks in our phylogenetic tree construction. So once we construct this tree, it sticks out pretty clearly that we have 2 unique clusters from New York. We have 1 unique cluster from the mountain area. And now we are interested, is our Nevada sample highly genetically related to these? And what we can conclude from this is that this is a distinct Nevada group. So while there may have been some transmission from New York to Nevada, this was not a direct transmission. The samples are simply not related enough. Candida auris is a slow evolving pathogen, so we could set that threshold very nice and tight, and we can see that there is enough genomic distance between the Nevada group and the New York group to say that there was a unique introduction event in Nevada, not related to the New York group necessarily. So from that, the investigators in Nevada were able to identify introduction events, and then they were able to go out and implement public health policy decisions to reduce the spread of these diseases through their population. All right, now we have a foodborne outbreak of salmonella. This is more of a clinical environment investigation. There were 7 epidemiologically confirmed foodborne outbreaks, 55 samples. They were all typed by pulse field gel electrophoresis initially, which is a legacy biomolecular assay that we perform in the laboratory to get an idea of the genomic relatedness between samples. Unfortunately, PFGE is not a very high resolution technique. While it is an amazing tool that has done a ton for public health and clinical microbiologists throughout history, next generation sequencing just allows a much higher level of resolution. So after PFGE was performed, samples were sequenced and a phylogenetic tree was constructed from the SNP matrix. And once we constructed the phylogenetic tree, this is what we saw. Now, quickly, green samples here are all part of the same PFGE sample pattern, and the red samples are all part of the same PFGE pattern. If you have not seen the first session of this Bioinformatics Fundamentals course, I strongly encourage you to go do so, where we talk about PFGE a little bit. For now, suffice it to say that unique PFGE patterns indicate highly related samples. So we can see here all of the green samples clustering together, all of the red samples kind of in the same clade. That's great. We have two distinct clusters. That is about the level of resolution that you can get with PFGE. You can see that these samples are related, all the samples in red are related. But then when we perform sequencing, we can really see the intricacies of these relationships. We can see that there are not just two clades, but many subclades within each of these clades. So you can see that the green clade up top is clearly divided into two smaller groups of highly related samples. Within the red samples down here, we can see many different outbreaks occurring over time. And if we think of this in a clinical setting, if you see the emergence of an outbreak in the year 2000, and then we see a second outbreak of a highly genomically related sample a year later, that to me indicates there may be some sort of contamination, some biofilm occurring in that setting that might be causing this recurring outbreak. There also might be a community reservoir of this outbreak currently circulating, or there might be a food product or some medical device where we can trace all of these outbreaks back to the root sequence, which is one of these inferred ancestral sequences, I'm sure, but we just don't have that sequencing data. So by seeing multiple outbreaks over time that are highly genetically related, we can infer something about the dynamics of these outbreaks. Where are they coming from? If we had additional epidemiological data, like, for example, the room in the hospital in which each of these individuals was staying, and we could relate them geographically, that might help us pinpoint the exact piece of medical equipment or the exact source of the biofilm from which the samples are originally stemming. So in comparison with pulse field gel electrophoresis, we don't have two groups of samples. We have many nuanced relationships depicted here between these samples with many subclusters within the clusters that were identified by PFGE. Again, kudos to the PFGE community. I have nothing but respect for that technique, but next generation sequencing allows for much greater resolution. So that brings us towards the end of this course. Thank you all so much for participating. The key takeaways here, we want to make sure that we understand how much genomic epidemiology has to offer us. Relative to the traditional molecular biology techniques, genomic epidemiology offers a much higher resolution look into the pathogens spreading throughout our communities. We must develop robust wet and dry lab techniques. There are many protocols out there for extraction, library prep, sequencing, but on the bioinformatics side, not a lot of bioinformatics developers produce protocols for their workflows. So reproducibility is super important when it comes to bioinformatics. Please make sure that you're running your bioinformatics workflow in a way that you're keeping track of every version of every tool used so that your work is reproducible. So in summary, genomic epidemiology is the combination of pathogen genomic data with traditional epidemiological data, such as geographic relatedness, to create a 1 + 1 = 3 super high resolution, super information rich phylogenetic tree. Phylogenetic analysis allows us to visualize the evolutionary relationships in a tree. We then use those evolutionary relationships as a proxy for the likelihood of transmission. And from there, we can construct a transmission network where we can start to pinpoint introduction events, individuals that are leading to a disproportionate spread of the pathogen through the community. And we can implement public health policy decisions, whether that be at the national scale or within a hospital ward. And then we went over a few case studies in public health just to give examples of how these approaches have been used in publications in the scientific literature. There is a ton more out there on this stuff. So if you're interested, you could read for days. But if you're interested in getting started with public health, pathogen genomics, or clinical microbiology, and using genomic epidemiology in order to enhance your ability to detect the spread of a pathogen through a community, definitely make sure that you understand how to run these workflows, whether that be on the command line, through a graphical user interface, or through a web tool. These workflows, these tools are going to be super powerful, especially now that every laboratory in the world has a DNA sequencer post-pandemic. We're going to see a lot more genomic epidemiology and bioinformatics in the future. So with that, that brings us to the end of today's lecture. Thank you all so much for participating. If you have any interest in this at all, please feel free to reach out to me. I am super passionate about bioinformatics, about genomic epidemiology. I'm always interested to learn more and learn what everyone is working on around the country and around the world. So with that, here is our support email. If you have any questions about anything I presented on today, please feel free to reach out there. Otherwise, feel free to reach out to me directly or the CEO of Theogen Genomics, CTO of Theogen Genomics, Kevin Libwood. So feel free to reach out to any of these contacts. We would be more than happy to help you with anything in your genomic epidemiology outbreak investigation, genomic surveillance, or to understand how to characterize pathogens more accurately. So thank you all so much again. I really appreciate it. And this concludes the second session of the Bioinformatics Fundamentals course by Theogen Genomics. Thanks all.